Within the cell theory, you have 3 different parts. Let's memorize it, folks, okay? Cell is the basic unit of life. All things are made up of cells. Cells come from cells. Like, you either know it or you don't know it. All right, when we get past that, we go right into the microscope. So if we are looking at the microscope, we have the transmission electron microscope, the scanning electron microscope, we have the light compound microscope, and then we have the dissecting microscope. If we are talking about the transmission electron microscope, we have to recognize this microscope is the best of them all. It has a 50 million times magnification, okay? That is going to give us the greatest resolution, the greatest clarity, the greatest magnification. So if we are talking about the best microscope on the market, transmission electron microscope, okay? Underneath of that, we have the scanning electron microscope. The scanning electron microscope can zoom in 2 million times. To put that into comparison, ours have 1,000. So like if you're comparing our light compound microscope to the electron microscopes, we get 1,000. We're so close. Okay, the light compound microscope, that's what we use in class. So think mitosis. If I said, hey, how can I look at the parts of mitosis in a cell? We're going to use what we have in our classroom. If I gave you a whole sample, such as like an eyeball, that's going to go underneath the dissecting microscope. So if you're in forensics, if you're in anatomy, those are the classes where you're really going to see the dissecting microscope come into play. Now, as I was kind of talking before, when it comes to the best of the best, okay, magnification. When it comes to magnification, you can see that the electron microscopes have much better magnification range than we do. But ultimately, we are still talking about the zooming in and the zooming out of an object, okay? But 50 million to our 1,000. All right, when it comes to focus, I guess focus, I should back it up, I should just call it resolution. Okay, so the resolution is talking about the focus on a microscope. The electron microscopes are able to do what we cannot. Where we have a coarse and fine adjustment that raises and lowers just a smidget, these microscopes have computer programs that help run them. So we can get a heck of a lot finer picture on this. Think like box TV, high def TV. High def. Box TV. Okay, now when it comes to clarity, I actually think all of the microscopes do a pretty good job with clarity. Because clarity, remember, you're talking about contrast and light. Ultimately, our light compound microscopes have good light power to change the adjustment. The difference when it comes to the clarity is the fact that a scanning and transmission electron microscope use gold flecks. Yes, that's right, you heard me, gold flecks to contrast the samples. We use Walmart food coloring. So like we're in the same category, obviously. All right, so there's your microscopes. I will tell you, on this test, there is no picture of a microscope. You are not labeling parts of a microscope. We don't have time in our lives for the parts of a microscope. Know what they do. That's ultimately where it comes down to. All right, moving right into, my friend, the prokaryotic cell. When we are talking about the prokaryotic cell, with the prokaryotic cell, there are 8 different things you need to know. And yes, I do still have sample pictures up front if you need them. 1, you need to know the flagellum. Okay, so flagellum is movement of the prokaryotic cell. Next, we have the villi. Okay? The villi help with small movement. Ultimately, the flagellum moves the cell. The villi help with small movement. They help with attaching to external environments, but they also help with conjugation. That's the big thing with the villi, conjugation. Villi are attached to the capsule. So if we are talking about the outermost layer, the capsule, okay? The capsule helps attach to environmental factors. Inside of the capsule, this blue layer right here, now recognize the test is in black and white. So like you can't be like, oh, it'll be blue and I'll be fine. It's black and white, okay? The blue layer is the cell wall. The cell wall helps with protection, structure, shape. In plant cells, the cell wall helps with dehydration. It helps locks in water, okay? We do not have a picture of the plant cell on the test, but the cell wall is on the test. Okay, then we have the cell membrane. So the cell membrane on the prokaryotic cell is the innermost layer. It is the closest to the cytoplasm. So just keep that in mind. It's the closest to the inside. The cell membrane and plasma membrane, you will see them intermixed throughout the whole test, like plasma membrane, cell membrane, same. Okay, semi-permeable, allows some things in and some things out. Next, you will have the cytoplasm. That is all the yellow. Now again, remember the yellow is not going to be yellow on the test. It is just going to point to as much of the yellow as I can to show that that is where the cytoplasm is. Cytoplasm is that semi-gelatinous material that holds things in place. Then you have all our little teeny tiny pink dots. And our pink little dots, just the dots, okay? Those are your ribosomes, protein synthesis. To wrap up the prokaryotic cell, you have your nuclear region. Remember, it is not a nucleus. Do not put nucleus, you will get wrong. This is not a nucleus. Prokaryotic cells do not have a nucleus, but they do have a nuclear region that holds DNA. Don't mix them up. Prokaryotic cells have no nucleus. Okay, when it comes to a eukaryotic cell, now remember, for eukaryotic cells, we are talking about animals, plants, protists, fungi. We just happen to focus on the animal the most, okay? So when we are looking at this, first and foremost, we'll start with the flagellum because why not? It's on the outside, okay? The flagellum has the same function. We are talking about the movement of the cell. Opposite of the flagellum, we are talking about the ciliatose, the cilia. Okay, the cilia help with movement of smaller particles along the outside of the cell membrane. The biggest example that I have really told everybody, think fallopian tubes, think egg. To get the egg down the fallopian tube, cilia are used to create a current to push the egg down the tube. Now, they are helped by the microfilaments. The microfilaments are the finer of the two, and they assist the cilia in their job. The mighty mitochondria, the powerhouse of the cell. They make the ATP. This is the site of cellular respiration. The lysosome. Now, recognizing obviously in random pictures we never know what circle the arrow is pointing to, in this cell that is on the test, we know that the mushroom-looking thing is the lysosome. There are right and wrong answers on this test tomorrow because we are using all of the things we've seen every single day. Okay? The lysosome. Now, when we get here, okay, we're talking about our little centrioles, our centrosomes. These assist in cell division. The microtubules that become the spindle fibers, those assist in cell division. Remember, they help connect and move your chromosomes where they need to go. Now, if there was a drug out there that damaged the formation of the microtubules, then cell division can't happen. Because what's going to help line the chromosomes up or split the chromosomes apart? Nothing. The microtubules are used for all of that. So if anything was to damage the microtubules, cell division stops. Moving on. Now, let's head over here to the control center of the cell. So we have the control center of the cell. It is the nucleus.
What does the lysosomes do?
It eats the bacteria. Lysosomes eat bacteria. The control center of the cell, the nucleus, okay? Surrounded by the nucleus, you are going to see a bunch of ribosomes. Those ribosomes are also on the rough endoplasmic reticulum. Those ribosomes are making your proteins. Those proteins are sent to the rough endoplasmic reticulum for modification, okay? So those proteins go to modify. They're being sorted into their little packages, modifying. Once it goes from the rough endoplasmic reticulum, it goes into the smooth endoplasmic reticulum. Recognize on the test that I just put ER. So like rough ER, smooth ER, just because I didn't want to type out endoplasmic reticulum. Okay? The smooth ER adds the lipids and the carbohydrates to your packages. We further carry along to the Golgi apparatus, okay? So when we get to the Golgi apparatus, this is where all of your packages are being sorted for delivery. Okay, so the Golgi apparatus, our big green blob, but again, colors won't matter come tomorrow. We are sorting everything out. Now, we are sorting them into two different vehicles. You are sorting them into a vacuole or a vesicle. The vacuole cannot fuse. It cannot fuse. It is not attached to the cell membrane. It is not in the cell membrane. There is a right or wrong answer tomorrow. The vacuole is not attached to the cell membrane. The vesicle is attached to the cell membrane. You can see that it is fused into the cell membrane. The cell membrane is the blue outer layer of the cell. Still semi-permeable. Cell membrane semi-permeable. Vesicle fuses. The last little cell part that we have, because we just point to random things on our model, this is the perioxisome. So the perioxisome detoxifies. Perioxisome detoxifies. So again, if we were doing a close-up of the cell, the flagellum, the nucleus, the endoplasmic reticulum rough, smooth endoplasmic reticulum, Golgi apparatus, vacuole, vesicle, perioxisome, mitochondria, microtubules, centrosomes, cilia, microfilaments inside, lysosome, cell membrane. Now, even though plants aren't picture-wise on the test, you do need to recognize the chloroplast, okay? So, not necessarily in its picture purpose, but you do need to know that the chloroplast is the site of photosynthesis, okay? The chloroplast is how plants get their energy. Okay. The next theory, because remember we have the cell theory and now we're talking about the endosymbiosis theory. When we're talking about the endosymbiosis theory, we have the mitochondria and we have the chloroplast. Here's the thing with the mitochondria and the chloroplast. They both have one thing in common, a phagocyte. Remember, phagocytes to eat. When we are talking about this, one phagocyte ate an aerobic bacteria. That aerobic bacteria gave rise to the mitochondria. That mitochondria is cellular respiration, coming up in Unit 3. Well, not all things eat the same. So, one phagocyte ate a cyanobacteria. That cyanobacteria gave rise to the chloroplast because it uses chemical energy, and that is where we get photosynthesis from, also in our next unit. Okay, that ultimately gets us to the intracellular junctions. For a lot of people, I recognize the intracellular junctions have been highlighted red. So when it comes to the intracellular junctions, the first one, the plasmodesmata, okay? When we're looking at the plasmodesmata, and I'm going to kind of just put a dotted line here, this is the only one found in plants. The gap junction, the tight junction, and the desmosome are all found in animals. Now with this, remember all of your intracellular junctions are between the cells. So whenever we're talking about an intracellular junction, we are talking about between cells. For plasmodesmata, it helps with transportation, and communication. Now that's for a plant cell. For us, our gap junction is pretty much the same thing in animals. That gap junction helps with transportation and communication. You then have your tight junction. Nobody wants their stomach acids leaking throughout their body. Okay, we need tight junctions. Essentially zip it up, waterproof seal that. Okay, so a lot of times your tight junctions create adhesive seals. So adhesion, okay? We are creating that tight bond. We don't want fluids leaking. Your last one is your desmosomes. This helps with cell flexibility. When we're talking about cell flexibility, it's like your skins. We've talked about your epithelial cells, so your skin. Okay, it allows your skin muscles to go out, but yet come back in. No one wants to stretch out their arm and then rip apart their skin cells. You don't. So, now that we're talking about cells, let's talk about that cell membrane. Your cell membrane is made of a phospholipid bilayer, okay? So within your phospholipid bilayer, you have hydrophilic heads and hydrophobic tails. This setup allows some things in and some things out. That's that semi-permeability. Now, I'm going to combine this with passive transport, just so everybody kind of knows. When we are talking about this, things like oxygen. Oxygen can go in, oxygen can go out. Oxygen does not need help across the cell membrane. It can just diffuse its way in and out. So something like oxygen is called diffusion. It does not need any help. However, we all recognize that sugar, glucose, is the key to making ATP and cellular respiration, and without eating, we kind of like die. So how does sugar get in? Because sugar gets blocked. Well, part of the fluid mosaic model talks about these proteins. And that protein channel allows things like glucose to get into your cell. Okay, so when things can't get through on their own, we have helpers. The proteins are the helpers embedded inside of your phospholipid bilayer to get bigger things out. Now, the other thing that can easily go back and forth is water. Okay, water uses osmosis to go in and out of the cell. Okay, that protein channel is facilitated diffusion, because facilitate, to assist, to help, facilitated diffusion. Okay, now there are a couple other things embedded inside of your phospholipid bilayer. We have cholesterol. Essentially, cholesterol is helping your more Arctic species from freezing and going solid. The cholesterol helps it maintain fluidity. Without the cholesterol, if you lived in subarctic temperatures, your body would freeze. That's a great way to die. The other thing you have is little carbs. And those are like your little receptors that send off signals that say, I need you over here. Like, come on, oxygen, you're needed here. Or, hey, NADPH, you're needed over here. So that kind of directs everything on where it needs to go. Now, I did say I was combining this. So, diffusion, osmosis, and facilitated diffusion. These are all passive transport. When we think passive, no energy. We go from high to low. This also leads to tonicity. So passive transport and tonicity went hand in hand. And that was talking about which way the water would flow in a solution. Essentially, this was our lab we did with the potatoes and the dialysis tubing. So if we were looking at that, and this came from one of your case studies. Okay, so all of the interior of your cells are going to have 10. In this situation, the solution on the outside also has 10. We'll go 30 on the outside of this, and we'll go 3 on the outside of that. Okay, so in the first example, water is going to equally flow out and equally flow in. Okay, so water is going to flow equally. This is going to lead to isotonic. When we look at this, follow the number. Okay, so the 30% concentrate is higher on the outside, so water is going to follow that to balance everything out. It's going to actually leave your cell. Because it is leaving your cell, causes it to shrink. That's hypertonic. Because it was higher on the outside. The last one, now that our cell concentrate is higher on the inside, it's lower on the outside, that is going to cause the water to enter, which will cause it eventually to burst. That's hypotonic, because it is low. your passive transports. Now, opposite of passive transport is your active transport. We know that to be active, you need energy. In this situation, we're going from low to high, okay, because we have to have the energy to go up the concentration gradient. In this situation, one example would be your sodium-potassium pump. It is imperative that you remember the numbers, okay? Sodium is not good for us, like in terms of salt. That's how I always think of it. I don't want salt. So 3 sodium, they go out. But potassium is in bananas, and bananas are good for our health. So we'll take 2 potassium in.
Wait, isotonic, hypertonic, and hypo, they're all passive transport?
Yeah, they're all tonicity dealing with passive transport. Because it's the movement of water molecules, that's osmosis. Okay, along with the sodium-potassium pump, exocytosis, endocytosis, phagocytosis, penocytosis, receptor mediated. If we are talking about exocytosis, think to exit. That is exiting our life. To exit. To release, get rid of, goodbye, so long, see ya. When we think endo, to enter. Phagocytosis, fatty the phagocyte, it eats, to eat. Penocytosis is so much more sophisticated, to drink. Receptor mediated is specific. It's got specific targets. Only specific enzymes can go to a receptor mediated. So, specific. Remember, the rest of the words on the test are meant to confuse you. That's really ultimately, when you think of multiple choice questions and matching questions, there's more than just one key word. I mean, right? What a great test if it was just to exit. But it talks in a sentence, and it talks about how we are releasing bacteria from a cell to exit. You have to find the key words in the sentences, because it's not just going to say, hey, come on in, to enter. So you really have to see through that. To engulf, to eat, to ingest, okay? I wish I could make it just one key word for the whole entire test. I really do, but I can't. Okay, this is all of chapter 3. We are now about to go into 6 and 7. Here we go. Okay. We're now talking about the cell cycle. When we look at the cell cycle, remember, your cells are currently sitting in this. Your cells are sitting in interphase. That is where they spend most of their time, in interphase. Okay? Interphase consists of G1, S, and G2. G1 is where your cells are growing and carrying out their normal functions. All of your cells have normal functions. They have different growth rates and different functions. So therefore, the amount of time they spend in G1 is a little different. Once your cells have grown to the right size and shape, they're going to go into DNA replication, where we replicate their genetic information. After we have replicated their genetic information, we're going to send them into another growth point. All of that is normal. This is where your cells spend most of their time. Now, if for whatever reason your cells aren't replicating, cardiac cells, nerve cells, they're going to sit in G0. G0, no cell division. Okay, it's like that waiting room. Now, there are certain situations in which cells will come out of G0. If we think about your liver cells go into G0 when it's too toxic. When the peroxisome detoxifies the body, then those liver cells can enter back into the cell cycle. Okay. Remember, there is a checkpoint in G1, there is a checkpoint in G2. Those checkpoints are making sure mutated cells are not going through. We don't want damaged cells leaking through the cell cycle, or they become uncontrolled cell growth. That can lead to tumors. That can lead to cancer. That can lead to abnormalities and death. Now, if everything is good to go, you are going to enter into the M phase. The M phase is where your cell finally divides, because mitosis and cytokinesis are where the cell starts to divide. In interphase, everything is just kind of randomly like chilling out, okay? We're growing, normal functioning. In prophase, we are starting to break down our nuclear envelope. We are starting to put things in their places until we get to our prometaphase. Okay, in prometaphase, our chromosomes don't go away. I recognize sometimes I don't draw them in my pictures, but they don't go away. Ultimately, we are talking about the connecting of our spindle fibers. So our spindle fibers are starting to connect to everything. Okay, when we get into metaphase, whether I draw 3 chromosomes, 4 chromosomes, 6 chromosomes, 100 chromosomes, it's all the same. They're going to line up in the middle. In anaphase, they are going to pull apart. Anaphase is the first place you are going to see chromatids. There was a chart in your lab that is due today that had like the 2N. Where do you see sister chromatids? Well, you see them first in anaphase. You'll see them again in anaphase 2, but you see them first in anaphase. We then get into telophase. That cleavage furrow is going to form in animals. In plants, you're going to get a cell plate.
Is this for meiosis or mitosis?
Mitosis. Now, in mitosis, we are starting with a 2N diploid cell. We are ending with diploid cells. Those cells are identical. Mitosis gives you identical cells, just like binary fission. Binary fission is how asexual prokaryotes reproduce. They reproduce with one cell becoming two cells, and it's all identical. Remember, asexual reproduction is great in regards to not a lot of energy. You don't need to find a mate. One cell becomes two cell. However, asexual reproduction is also kind of a bad thing, because if one thing takes you out, it takes out the whole entire community. You all die. Sexual reproduction, meiosis. Let's talk about meiosis. When we talk about meiosis, we are still starting with 2N. Okay? Ultimately, all of your sex cells are in sex organs. Organs are body cells. They have to be diploid. So you start with diploid. We start with interphase. In prophase 1, the hard part for tomorrow's test is going to be seeing the different colors, because it's a black and white test. I'm going to do my best for you, okay? I'm going to do my best for you. Now, if we were going off to the side, prophase 1 in meiosis is important because it has crossing over. Crossing over helps with genetic variation, so crossing over is important. It only happens in prophase 1. Okay, prometaphase, really they still line up. In metaphase 1, you are going to notice that the chromosomes are in pairs in the middle. Okay, so these are still homologous chromosomes. These homologous chromosomes don't actually separate until anaphase 1. So in anaphase 1, they pull apart. This is unique. On your lab, it said in a chart, where do homologous chromosomes separate? Anaphase 1. Homologous chromosomes separate. They don't separate anywhere else, okay? We then go into telophase 1. You will see there's still chromosomes, okay? Until we chop them apart in cytokinesis 1. Now, I recognize for some of you, the math doesn't work in your mind. It's okay. Don't stress over it. Just know that these cells right here are haploid. You just made your first haploid cut, okay? Because they are half of what we started with. Now we go into prophase 2. And I'm going to draw prophase 2 right next to this so you can see there's no break. Interphase doesn't occur again. This is happening in 2 cells. We're going to go into prometaphase 2. These are your phases in which you are going to start to recognize mitosis because they look so similar. Metaphase 1. They are now lined up on top of each other in the center. I apologize, my board is only so big, so like I have to carry us over here, okay? Please know that if my board was even bigger, I would just keep going in a straight line. Anaphase 2 is now where you see them get pulled apart. So here, right here, you have chromatids. You see chromatids in mitosis anaphase and anaphase 2 of meiosis. Telophase 2. Which leads us to our final division. Just like my picture is not even and unique every time I draw it, we have 4 unique haploid cells. That's how we make sex chromosomes.
Wait, I thought mitosis was sex cells.
No. Mitosis. Mitosis. I'm making toes. Mitosis makes my toes. My body cells. Again, seriously, if all of a sudden in the middle of test tomorrow, I start seeing people go, mitosis. Great, you make your damn toes in mitosis. Make your body cells, okay? We don't talk about meiosis. Nor do we do meiosis in this classroom. That's how the couch gets thrown away. All right, I'm erasing this, by the way. We increase genetic diversity in crossing over. We increase genetic diversity in random assortment. Because I could draw this picture 4 times in a row and draw it different every one of those 4 times. Which I have been for 3 days in a row. Okay, this gets us all the way through 6 and into 7. So let's keep talking about 6 into 7. If we are talking about sexual reproduction, do all organisms have the same sex chromosomes?
No.
Nope, they don't. We have XX and XY. Birds have ZZ and ZW. Bees have diploid dominant, obviously. And then they have haploid. Sorry, gents. If we are talking about the fact that not everything has the same sex chromosomes, it would only make sense that not everything would have the same sex life cycle. So, the life cycle for all of them is going to start off with a zygote. Oh my God, that's my Thank you. They're leaping through the air like dance class. Okay? We don't have time to draw pictures of people dancing, okay? We just have time to label things.
Label that for the test.
We actually, like when I say label, I mean like this is going to be an animal which turns into diploid dominant. We don't have time to fill this in. So like, hang tight. You have to be able to recognize that this picture is diploid dominant. It spends most of its life cycle in diploid, okay? And that it is an animal. That's what I mean by label this cycle. We're barely gonna have time to finish. We don't have time to color pictures, okay? Again, mitosis, that's my plant. It's been a long day already, clearly, okay? There's kind of a trick to these pictures, especially knowing that I drew all of them and then put them into the test and like I drew them. So far you'll notice with animals, we are dominant, obviously. The animal kingdom is dominant, okay? Therefore we spend most of our time in 2N. We actually only undergo one meiotic division. So like we really only grow once, okay? Plants. Plants are 50-50. We need plants, but plants need us, okay? It's like a circle of life. So when we look at this alternation of generation, you're going to see it's 50-50. They actually have mitosis twice, okay? So 2 mitosis for 50-50. When we get into fungi, which I'm starting to learn how like bleh fungi are, okay? So fungi have a period of growth that goes right into meiosis. That meiosis still gives them the spore. Well, that spore has to grow. So here's our second growth period. Why don't we just call that mitosis? Fungus. I mean, right? That's why they have to be unique. That leads us into our gametophyte or our fungus. Well, guess what? That still needs to grow. So they essentially have 3 growth periods before they ever get to their gametes that of course then need to fertilize and then become a zygote. So if we look at this, most of their life cycle is spent in haploid. Well, at this point I have determined that fungi are just half when I chop them up. They're not as dominant as I am because I have the knife and they don't. Again, you have to be able to recognize these with things missing from the pictures.
Saying, what's the second one?
Alternation of generations. It alternates, okay? So think 50-50, it alternates. Okay, the last part of all of this. It's the last part of 7.3. If you have aneuploidy, okay, so aneuploidy is a mistake in your chromosomes. All right, that would be monosomy where you might have 1 chromosome, or it could be trisomy where you have 3 chromosomes. If you have polyploidy, you have multiple sets of chromosomes. Okay, so instead of just adding like 1 or not having 1, now you have a set, so you have 2 added or 4 added. You have extra sets in polyploidy. Deletion, to remove. Duplication, to add. Translocation, to move from like the front to the back, or the front to the middle, or the middle to the end. It's, again, think location, location moves, to move. And then you have inversion, to come out, flip, go back in, to invert.